All right, so we are here to talk a little bit about C4 plants. And so if you'll remember, C3 plants were called that. Whoops, wrong thing. C3 plants were called that because when they do, I got this zoomed in really far, so I don't know if I can see it all. When they, after rubisco, they do that carbon fixation, the first stable molecule is this 3-carbon. And so that's why we call them C3 plants. But unfortunately, C3 plants have this problem of photorespiration, where rubisco sometimes binds oxygen to the RUBP. That creates a toxic 2-carbon molecule, and that's bad news bears for the plant. So this is what a normal leaf looks like, just to get you kind of used to the structure again. These are all mesophyll cells here in green, okay, and they're the ones that are doing photosynthesis. So lots of thylakoids, lots of chlorophyll, all that. This is your vein. Your vein or vascular bundle, you may hear it called, is going to be made of xylem and phloem. Xylem brings water up from the roots to the leaves and sugar down from the leaves to the roots for storage. We have an upper epidermis, so this green line, and we have a lower epidermis. This kind of highlighted is representing a cuticle. It's a waxy layer, so it's pretty much clear, so sunlight can get through, but it protects the plant from desiccation or from drying out. It prevents water from evaporating. And then the last thing we have are these stomata. And stomata are for gas exchange. So we're going to see CO2 going in, and we're going to see O2 coming out, okay? Because the plant's constantly producing oxygen, it wants to get rid of the excess, and it needs CO2 for those light independent reactions. On hot, dry days, another thing that you might see slipping out of this is water vapor, and that's bad because the plant can dehydrate. So what they're going to do on hot, dry days is they're going to close up all their stomata. That's great. It prevents water loss. They don't dehydrate. However, it also prevents this gas exchange, and so it's going to prevent the plant from getting rid of all that excess oxygen, and it's going to prevent the plant from bringing in carbon dioxide. So what you're going to see over time so what you want in a normal situation is you want lots of CO2 and little oxygen, okay, because that's where rubisco is going to do the right thing. But when you close the stomata, so now the oxygen can't escape, okay? Sorry about this, so zoomed in, I keep forgetting. You're going to see your oxygen levels rise because the oxygen that you're producing in the light-dependent reactions can't leave. And you're going to see your CO2 levels drop because you're using up what CO2 you have in the light-independent reactions. And so over time, you're going to see those levels, instead of being what you want, they're going to flip-flop. When they flip-flop is when rubisco is going to start grabbing these instead of these because there's just more oxygen available, and they'll start doing that photorespiration, and that's not what we want, okay? So one evolutionary change that has occurred is the production, or the, I guess you could say the mutations, I guess, that led to a C4 plant. So I'm just going to give you an overview of the leaf. So we have, again, our upper epidermis. We would have our cuticle up there. We would have our lower epidermis. Kind of ignore this down here. I'm not ready to talk about that yet, so we'll hide that. We have our lower epidermis with the stomata. Okay, so those are those stomata there for gas exchange. They're open right now. And instead of having just mesophyll, they actually have a cell around their bundle, their vascular bundle or their vein. They have a set of cells called the bundle sheath cells. Now, to sheath means to closely wrap. So when I think of sheath, I think of a sword. If you carry a sword on your hip and you pull it out, what you're pulling it out of is the sheath. So the sheath is the kind of leather thing that the sword is in, but it's tightly, it's not much bigger than the sword. So when we say this bundle sheath cells, it is sheathing. So it is closely wrapped around the vascular bundle. That's where it gets its name. I was going to abbreviate it, but the abbreviation would be BS. I didn't feel like that was a good idea, so that's why I didn't abbreviate it. All right, and then around the bundle sheath cells, you have your mesophyll cells, okay? You still have your palisade mesophyll up there. You still have some spongy, which I'm really bad at drawing those. They should look like amoebas. But anyway, your spongy mesophyll. So all of the green ones are mesophylls. But the big thing to note is that your mesophyll cells are surrounding your bundle sheath cells. That's keeping your bundle sheath cells from being exposed to this air, and that'll be important in just a second. All right, so what we're going to do is we're going to kind of zoom in on like this area. So we're going to have a little bit of vein, we're going to have a bundle sheath cell, and a mesophyll cell. So that's what I've got down here, okay? What C4 plants do and why they're called C4 plants and all that good stuff. What they're going to do is they are going to do gas exchange, and I didn't draw here, but remember there's more bundle sheaths and there's more mesophills. So this does not have access to air. So what we're going to see is we're going to see our CO2 coming in here. We're going to see the light-dependent reactions happening here. And if you'll remember, they're producing oxygen, ATP, and NADPH. They're going to produce oxygen, which is going to leave, go right into this space, and go out of the stomata. Okay, I don't have a stomata drawn there, but you can get the idea. And so that's going to keep oxygen up here in the mesophyll cells. Okay, I am going to abbreviate the bundle sheath cells, but I'll put a C. So mesophyll cells, bundle sheath cells. Meanwhile, we are going to do the light-independent reaction, or the Calvin cycle, down here. And if you'll remember, that is where rubisco is. So what these plants have done, these C4 plants, is they have basically said, oxygen and rubisco, you cannot behave when you're together, so we're just not going to let you ever be together. So rubisco is going to be down in the bundle sheath cells, oxygen is going to be produced in the mesophyll cells, and then it leaves, and so they never run into each other. Okay, well that's great and all, but how does the CO2 get down here? Well, the CO2 is going to be joined with a 3-carbon molecule called PEP. Okay, so the CO2 gets joined to PEP. That's going to form a 4-carbon molecule. The enzyme that does this is called PEP carboxylase. Carboxylase. And that simply means an enzyme that adds a carbon oxygen or carbon dioxide to PEP. Okay, so the enzyme that does this is going to be that, PEP carboxylase. It adds this CO2 to the PEP. That makes a 4-carbon. That 4-carbon comes down into, it's shuttled down into the bundle sheath cells. It's going to drop off that CO2, which takes it back to a 3-carbon that then goes back up. So it's almost like a taxi cab or an Uber. It picks up that carbon dioxide, shuttles it down into the bundle sheath cells, drops it off, and then leaves to pick up another one. So your I can statements are I can draw and label the structure of a C4 leaf. So hopefully you can draw either this or the zoomed in version down here. I can explain the process C4 plants use to isolate rubisco from oxygen and prevent photorespiration. They're going to use a physical separation, so they are physically in 2 different locations. Oxygen in the mesophyll cells, rubisco in the bundle sheath cells, so that they're not in the same place at the same time. And I can explain the role of PEP, which is the Uber, okay, and PEP carboxylase, which is the enzyme that puts CO2 into the Uber, into the taxi, so that it can shuttle it down here. So I can explain the role of PEP and PEP carboxylase. PEP is going to shuttle the carbon dioxide down here and drop it off. PEP carboxylase is going to join PEP and CO2 together. And so that's how C4 plants try to avoid photorespiration. They do a really good job of it, and it allows them to live in hot, dry environments. And so you often see these C4 plants existing in a hot, dry environment because they are able to outcompete other species that are there. So because they are avoiding photorespiration and they're not wasting energy, they are able to survive and be healthier than, say, a C3 plant in a hot, dry environment. And so you will see them more commonly in those places. And that is that. So I hope that helps you understand C4 plants, their structure, and how they use that structure to prevent photorespiration and save them some energy and increase their chance for survival. But if you don't understand anything, or if you still have any questions, let me know.